first,
No,
first
Joanna.
of all, okay. Thank you, Elena. First of all, I would like to thank Leo for accepting to provide this seminar. And maybe with no further delays, we already have a lot of attendees, participants in this meeting. So maybe I can start by introducing Leo, and then afterwards you can proceed, of course. If you like, I guess you can already test if you can share your screen.
I will.
Might be helpful. Oh, perfect. In the meanwhile, I will then present our speaker. So it's Leo van Grunsven. We had the chance to meet already earlier in another context. But it's of course my pleasure to introduce you, Leo. Just a short introduction. Leo obtained his PhD in Lyon, France. Afterwards, he moved to perform a postdoctoral training at NIH, and then moved back to Belgium to KU Leuven, and then to the VUB University in Belgium as well. Since 2009, he leads the liver cell biology research group. His group, the studies performed at his group focus mostly on understanding the molecular mechanisms involved in liver, such as homeostasis, fibrosis, and regeneration, with a special focus on hepatic stellate cells. His group was the first to identify autophagy, AGE, and hyposignaling as key mechanisms involved in hepatic stellate cell activation during the liver fibrogenesis. They also established the first hepatocyte injury-dependent in vitro liver fibrosis model by using spheroid cultures of human hepatocytes and hepatic stellate cells. The current research efforts include the development of more advanced in vitro systems for chronic liver disease using primary mouse and iPSC-derived liver cells, and investigation of stress pathways in hepatic stellate cells and other sinusoidal liver cells during the acute and chronic liver injury. And so today, his talk, or this seminar, is entitled, as expected, Liver Organoids for Acute and Chronic Liver Disease Modeling from Primary Cells to Stem Cell-Based Approaches. So Leo, thank you so much.
Okay.
The floor is yours. Thank you so much.
Thank you, Joanna, for the introduction. I will put a pointer, although I might not use it a lot. Thank you for giving me the opportunity to talk to all these people. I want to get rid. Okay, there it is. And I'm going to talk to you about the work we've done on liver organoids for acute and chronic liver disease modeling. I'm from the VUB, the Vrije Universiteit Brussels. So this is in Brussels. Here we have the center in the back, and here at the front we have our campus with the University Hospital, which is the biggest and tallest building, and our lab is here located in one of the many research buildings here on this pretty green campus if I look at from the sky. We've been interested in hepatic stellate cells ever since I joined the lab, because my predecessor, he was one of the first to isolate hepatic stellate cells from rat, actually. It was Professor Geertz. Now, it's a very short introduction. Many of you know that it takes many decades to go from a healthy liver to a fibrotic liver and a cirrhotic liver. We all know what's causing it: hepatitis viruses, steatotic liver disease, alcohol, and drug abuse. And although there's now recently there has been one drug on the market that also shows very promising results as an antifibrotic, there were no good efficient therapies available for many years. So there's about 150,000 people dying every year in the EU of a chronic liver disease. So why do we need liver organoids? And this has to do with, well, of course, we have to test new medication, not even just a liver, anti-fibrotic medication, but any kind of medication needs to be tested for adverse effects on hepatocytes or other cell types in the liver. We would like to find new targets that could be used as antifibrotic drugs. So maybe liver organoids would be a good way of studying this. And the problem is so far, or at least, now I'm lying because these slides are already a couple of years old. The in vitro models used in many companies were very basic, like using 96 well plates of, for instance, HepG2 cells, and this does not, of course, reflect the intricate system of the liver. And then last, the preclinical models that we use, like mouse and rats, that we put on high-fat diets or alcohol, etc., are not really resembling humans. So having a human liver organoid model would also be advantageous, I think. And last but not least is that we, of course, would like to reduce the number of animals used for in experiments. And this is data from the Allures database, where in 2019, 4.5 million rodents were used for translation and applied research, of which almost 700,000 were used for gastrointestinal system and also liver tox. So reducing the animal number would also be nice. And organoids is one way to do this. So just to get something straight, what is an organoid? It's three-dimensional, miniaturized, simplified version of an organ or tissue grown in vitro from stem cells or tissue-specific cells. And I will talk about how we approach both of these. And then they should or are designed to mimic the structure and function of the organ or tissue in the human body to some extent, or at least mimic some of the function of this organ. For the liver, this means that we would like to have at least, the liver cell types like hepatocytes, stellate cells, LSEC, Kupffer cells, or even cholangiocytes in such an organoid model. Preferentially, we would like to have a 3D structure in there that kind of resembles the liver, although we're far away from that. It should have some functionality from the liver, and preferentially it would be, reproducible as well. Cherry on top would be that it would also be patient specific, so that we could maybe find specific cures for people with specific SNPs or mutations. So a closer look at what we would like to make is a liver lobule. Liver is made up of hundreds of thousands of liver lobules, as you know, and in which the blood flows from the portal vein to the central vein through liver sinusoids, which are made of LSEC, which are specialized endothelial cells which have fenestrae, which allows the passing of proteins, but also fat, etc., to the hepatocytes, which metabolize can metabolize many compounds, but also produce things like albumin, for instance. Besides that, we have the stellate cells, which are in the space of this, so the area between the LSEC and the hepatocytes, which maintain the extracellular matrix balance and also keep structure to the liver. In the end, this healthy liver architecture is disturbed when we eat unhealthy or drink too much. We get damaged hepatocytes here. This leads to an inflammatory response, also recruitment of monocytes that turn into activated Kupffer cells. We get activated stellate cells, and in the end, high extracellular matrix production. We can mimic this easily, and as many are doing still, is we can mimic this in the lab by just giving a mouse 4 or 8 or even more weeks of CCl4, carbon tetrachloride, which is metabolized by the hepatocytes, but then becomes toxic to them. And this toxicity leads to scar formation here, indicated in red by serious red staining of cross-linked collagen. If you would isolate stellate cells from these livers, they would show high expression of alpha SMA, collagens, and loxels, indicating that activated stellate cells are present. And actually, activated stellate cells, they are the main producing collagen or extracellular matrix producing cell type in a damaged liver. So to study mechanisms, 40 years ago, they started isolating quiescent stellate cells. So from a healthy liver, put them in culture, and they are these tiny cells with large lipid droplets filled with retinoids. Over time, in 10 days, they become very big cells, myofibroblastic, and they also express high amounts of alpha SMA, collagen, and loxels. So some of these aspects of this in vitro activation actually resembles what happens in vivo. And this model has been used many, many years to study mechanisms of fibrosis. Now, we isolate them in a lab from mice. Many people have been doing it from rat. You can do it from mice as well. You perfuse, we do it in vivo perfusion. We take out the hepatocytes by a Percol gradient, and the leftover non-parenchymal cells we put in a fax, and we can sort out the UV positive cells because of their retinol esters, and we can put these in culture. They're very pure cells, all desmin positive. No other cell types are really in these sorted cells. You can do an 8% nicodens, so a gradient centrifugation. It leads also pretty good stellate cells, but a bit less pure, and you need more mice to do it. Now, life is not 2D, it's 3D, because in 2D, if you plate these cells, they activate. And this is because the surface is very rigid, which leads to stellate cell activation. And we found this out because we did a study in 2015 in which we studied hyposignaling. So in a 2D culture, so regular culture dishes, the surface is rigid, hyposignaling is off, and stellate cells activate. If you put them in spheroids directly, hyposignaling is on, and stellate cells remain quiescent. And they stay quiescent for a long time. You can see it here in this figure, looking at alpha smooth muscle actin, so ACTA2. In 2D, stellate cell upregulate this activation marker of stellate cells highly. After already 4 days, it goes up immensely, but in 3D, this is not the case. It doesn't mean it's not upregulated. It's still like 10-fold upregulated, but a lot less. And you can keep these stellate cells for a while in 3D. And this, and we showed this in this JAP paper in 2015, is due to YAP in 2D, YAP downstream targets, CTGF, and anchored are highly upregulated, while in 3D, this is not the case. So does that mean that we can just disregard the entire activation in 2D and go for stellate cells in 3D? Would this be a better model? Well, we figured out also in 2015 that this is actually not the case, because what is missing in this model is these damaged hepatocytes. They are not there. So a bit later, we set out to make a culture. And for those who've already seen sometimes this presentation, I'm sorry, but this is still the animation I use. We use differentiated HepRGs that we got from BioPredict. We put them together with human primary stellate cells that we isolated from donor tissue from the clinic here. We passaged them a couple of times, and we put them together. These spheroids are roughly 200 micron in size and 2,000 cells. If you open them up, the stellate cells are in the center, and the hepatocytes are surrounding them. And if you do the staining, it looks like this for CYP3A4. There's roughly 20% of CYP3A4 positive cells in there, and the core is made of primary human hepatic stellate cells. What we also noticed that while primary human hepatic stellate cells, if you passage them, they're activated, but once you put them together with hepatocytes, they become quiescent, most likely because they just do not touch the 2D hard surface anymore, but they're in suspension, and they seem to like the functional hepatocytes. In this paper in 2016, we showed that if you damage these HepRGs, you can activate the stellate cells, and we could see an upregulation of collagen and loxels. We then went on to show that we could take away these primary human hepatic stellate cells, and replace them by human induced pluripotent stem cell derived stellate cell-like cells. And I use stellate cell-like cells because they're not, of course, not yet stellate cells. We used a rather short differentiation protocol that leads to these more or less cuboidal cells, but we usually passage them once or twice, and we then get a stellate cell-like phenotype, which looks like this, and they resemble a lot the primary human hepatic stellate cells that we have in culture. Large cells with a myofibroblastic phenotype. If we look at their RNA-seq of these IPS-derived stellate cells, you can see here IPS, and over time, during differentiation, they resemble more and more the primary human hepatic stellate cells that we can isolate from donor tissue. They're not similar yet. There's quite some variation here, but there's a trend towards an hepatic stellate cell. If we try to look at the functionality, we can see that they express stellate cell markers like alpha-SMA, collagen, desmin, PDGF receptor. You can induce activation by giving them TGF-beta, PDGF, BB, and LPS. And they also store vitamin A in their lipid droplets. The proof in the pudding is that we can make similar HepRG and IPS stellate cell spheroids. And if you have control conditions, you have very little serious red staining, some collagen, because there's always some collagen present, of course, in these spheroids. If you then expose them to acetaminophen or APAP, which damage the hepatocytes, you can see that this also leads to increase in serious red and also an increase of collagen, in these spheroids. We showed this for several other damages, and of course you can induce them as well in these spheroids, also directly induce them with TGF-beta. We can take the medium from these spheroid cultures, and you can see that in both cases, IPS stellate cells or primary human hepatic stellate cells, if you challenge them with APAP, and procollagen in the medium also goes up. Similarly, between those two cultures. So we have a good culture of an hepatocyte-like cell and an hepatic stellate cell-like cell. Both HepRGs, let's say, is the basis, but then we went back, and I won't present you that data, but we also went back to the mouse and isolated mouse hepatocytes and mouse hepatic stellate cells. And in all these three culture conditions, stellate cells remain quiescent once they are in these cultures with hepatocytes. The hepatocytes stay functional over a long time, and the stellate cells activate upon hepatocyte injury. This was, of course, very nice at that moment, but what we wanted to do is to have a more complex culture. So we would like to go for either primary mouse hepatocytes, stellate cell, Cooper cell, and LSEC, or IPS-derived cells, generating also 4 cell type co-cultures. I'll start first explaining what we did on the mouse cultures. We developed a protocol in which we inject with a secondary antibody. We inject mice with LX647, a secondary antibody. This is taken up by LSEC and by Cooper cells, but not by stellate cells. So if you make a single cell suspension of these livers after 2 hours after injecting, you can sort out these cell populations, pool them with hepatocytes that you can isolate from the same liver, and put them in ultra-low attachment plates. And you get these spheroids that quickly form after a day or two. Here, 4 days spheroids. They're, again, roughly 200 microns in diameter. There's no necrotic core present. And you can grow them up to like 14 days without a problem. If you look at these spheroids, you can see that all cell types are really crisscross through this spheroid, not like the HepRG and the primary human hepatic stellate cells. This is most likely because these cells are all freshly isolated. There's not one cell type that aggregates more quickly. So it's really a random architecture of different cells in these spheroids. And you can see again that if you use the stellate cell that you isolate here, you put them in 2D, they activate really quickly. But in these liver organoids, alpha-SMA and collagen stays really stable over time for a period of 15 days if you culture them correctly. We look closer to these spheroids, and with SEM. And if you look at the outside, you can identify really easily microvilli and also some biocanonically. If you crack it open, you can really see nice biocanonically that are formed in these spheroids. And even there's some fenestrated LSEC, which are easily detected at 4 days. At 7 days in culture, it's less easy, but they're still detectable. It's not as nice as if you would image a mouse liver freshly taken from a mouse, but still, this was one of the first times that we could see such nice fenestrae in an in vitro culture model after 4 days in culture. These spheroids behave quite well compared to 2D cultured. Well, of course, it's not very fair because these cells are 2D cultures and kept for 2 to 3 days in 4 days actually in a regular medium. Similar medium, but then in the spheroids, we see a high albumin production. It decreases over time, but it's still pretty good after 14 days. We can damage the hepatocytes if we add APAP again. You see it here as a decrease in ATP levels. And this APAP exposure, and this is, so I think 48 hours, we can see that there's also an increase in collagen in these spheroids. Same thing, if we take the medium out of these spheroids, and this time it's collagen 6, we can see an increase in collagen 6 in these spheroids after APAP exposure. Now we can use these spheroids for many things. So one thing is just to look at direct activation, and then TGF-beta is the easiest. TGF-beta activates these stellate cells. So in these spheroids, this is also the case. High upregulation of ACTA2, COL, and COL6A3. But what is more interesting and that we actually should look more into is that you can look at what, which chemokines, growth factors, molecules affect which cell type. So here as a case study, we used LPS, and we know that LPS can activate Kupffer cells, but also has an activating effect on stellate cells. So we did 2 setups, one spheroids with all 4 cell types and a spheroid without Kupffer cells. And if you then look at the secreted proteins, you can see that for TNF-alpha protein to be upregulated, you need and LPS and the Kupffer cells, because without Kupffer cells, there's no upregulation of TNF-alpha. But for MCP1 protein upregulation, you actually don't need the Kupffer cells because the upregulation of MCP1 is as high in the absence as in the presence of Kupffer cells. Again, IL-1 beta is only upregulated in the presence of Kupffer cells and LPS. And totally, you need also Kupffer cells for the highest activation of stellate cells. So Kupffer cells plus LPS lead to the highest upregulation of collagen 1A1 in these spheroids. So if you then look at a scheme, you could think that LPS acts on both stellate cells and Kupffer cells. The Kupffer cells secrete TNF-alpha and IL-1 beta, and this leads to further activation of the stellate cells. So we used again the same spheroids again, and we just exposed them to IL-1 beta and TNF-alpha, and you see a nice upregulation of these of COL1 in these spheroids. Which means that we could really play around. We could also look at the activity of LSEC, whether they play a big role or not, but we haven't gotten around to do much of these studies, unfortunately. We could also mimic steatotic liver disease. And we did this by exposing the compounds through free fatty acids, oleic and palmitic acid in this case, for 10 days. And you see a nice accumulation of lipids in these spheroids. And if you add compounds that are have been tested to block lipid uptake, like for instance, elafibranor, and obeticholic acid, you can see a decrease in lipid uptake compared to, for instance, a treatment with selonsertib, which is an ASK inhibitor. We also asked ourselves, well, can these methyl D compounds block fibrosis? Because of course, if you add the compounds at the start, then you will not get steatosis and hence no fibrosis. So what we did is we developed a fibrosis model in which we add free fatty acid and LPS. And without the LPS, you get a tiny bit of fibrosis, but with the LPS, you see here a nice accumulation of collagens in the spheroids. If you then add that the last 48 hours, so there is steatosis in all the spheroids in the last 48 hours, that does not matter much. But if in the last 48 hours you add the same compounds again, you can actually see that mainly lanifibranor is able to block stellate cell activation in these steatotic liver spheroids. So why don't we use, this was all the mouse part. We also were interested for a while in primary human liver organoids, and there has been some studies that have done this, and they're listed here below. It's not that easy to do because not all primary human hepatocytes form spheroids, but what people usually do is that they mix non-parenchymal fractions with other hepatocytes, etc. And they can form spheroids. And there are some really nice studies that you can mix these primary human hepatocytes with NPCs, and you can also induce free fatty induced steatotic liver cells in these spheroids. But in this study here, for instance, you can see that the induction of collagen 1A1, already the basal level is really high in this combination of primary human hepatocytes with MPC1. MPC2 is the same. It actually goes down a bit when you treat with free fatty acids. It's actually only in this, in 1 out of 4 combinations that you see a nice induction of collagen in these spheroids. So although we think primary human liver organoids are the best, but it's very difficult to do because the reproducibility and also the quality of the NPCs in hepatocytes is very difficult to predict. So a solution here is to come with human iPSC-derived liver organoids. And together with Catherine Verfeilly from the KU Leuven, here also in Belgium, we set up another system to make 4 cell type organoid cultures. So in a previous paper from the lab of Verfeilly, they developed transgenic or modified iPSCs where they overexpress HNF1 alpha, FOXA3, and PROX1, then carried out a differentiation protocol, and you get to hepatoblast at day 8 of differentiation. And this is just a staining of HNF4 alpha for these cultures. You can re-optimize this iPS-derived stellate cell differentiation together with Pau Sancho Bru from Barcelona. And you get these cells that are alpha-SMA and vimentin positive. It's a heterogeneous population as well. And actually, Pau Sancho Bru showed in the last presentation that actually there's more like 4 or 5 different cell types in there. We, in the meantime, set up a system where we can induce endothelial cells by overexpressing ETV2. And so after 4 to 14 days, you get endothelial cells, again here stained for CD31. And then last, we use a protocol that is optimized by others, where you make embryo bodies, and by keep on culturing them, there's monocyte-like cells that are kind of budding off these embryo bodies, and you can collect them over multiple weeks and use these as monocytes. Or if you plate them in 2D, you can also kind of push them towards a macrophage-like phenotype. We put all these together, and this is some organization because one has to be, cultured for the day 8 days, the other 14 days, the other 2 weeks, the other 5 weeks. But then we can put these together, again, either in ultra low attachment plates, or we could put them in gels that are functionalized with extracellular matrix proteins and cell adhesion molecules. So I'll first briefly go into this, and then I'll talk a bit about this as well. This is only recently accepted in biofabrication like 2 weeks ago. So we played a lot with these spheroids. Why did we go for spheroids? Because it's easier than in these gels. Spheroids you can grow in 96 well or 384 wells. We put those hepatoblasts together with the other cell type, and we keep on maturing them over 21 days in culture. We played around with different ratios because in the past, in the previous papers that we published on this, at least with the mouse, they always questioned, well, you don't really use a good ratio that is present in the liver, because in the liver, actually, this ratio exists like 0.4 stellate cells versus 2.8 hepatocytes and 0.4 endothelial cells and 0.3 macrophages. Well, we use this ratio, 2.2 stellate cells versus 1 hepatocytes, and then a bit less endothelial cells and monocytes, macrophages or monocytes. We also did a co-culture where we put everything equal, so 1, 1, 1, and again to get into a total of roughly 2,000 to 3,000 cells. We optimized some medium conditions, but we also looked whether they would all form the same. And you clearly see here that the in vivo condition, it's very difficult to get good spheroids to form if we use the more hepatocytes than stellate cells in the rest, at least if we use our culture conditions. Equal, they form, but the best are these higher ratio of stellate cells, because probably the stellate cells really form a glue, producing quite some extracellular matrix between the other cell types. Surprising to us is that if you look at here, so these are RNA profiles of CYP3A4, PDGF receptor beta, CD68, and CD31, last one for endothelial cells. Any of these conditions, actually the cells mature similarly. At the start, there's maybe a bit of difference depending on how many cells or stellate cells you put in. But in the end, you end up with more or less the same level of maturity for hepatocytes, still lower than primary human, freshly isolated human hepatocytes. Stellate cells are more or less similar as well compared to human hepatic stellate cells. Kupffer cell markers are quite below the freshly isolated human Kupffer cells, and LSIC markers as well below the human LSIC isolated. But surprising to us is that actually, even though you don't form nice spheroids, the cells mature in the same way, which probably means that it's not the ratio that makes them differentiate that much, or the cell attachment is more the things that they secrete. We have the first couple of days, we have a bit of proliferation, but in the end, we don't have much proliferation anymore. These at 21 days, you have PDGF receptor positive cells and some CYP3A4 positive cells, some endothelial cells, and we can still detect some CD45 positive cells. We sequence these spheroids as well, and you can see that the different hepatocyte linked genes on phase 1, 2, drug metabolism, bile acid metabolism, and gluconeogenesis are upregulated between the hepatoblast stage and the 21 days later in this co-culture. Interesting as well is that activation markers of stellate cells like the collagen 5, the 6, the 1, they all drop. So again, the stellate cells, they become more quiescent once they are with the hepatocyte, and some quiescent markers like LRAD and COLOC10 go up. Endothelial cells and macrophages move less than the other cell types, but still some markers like PCAM or STAP1 go up, although like, for instance, STAP2 goes down over time. If we test these spheroids for drug-induced fibrosis, you can see that if we add APAP, their IC50 is not great. It's an IC50 of 28. With HepRGs, we were around 10. It's not great. We now have culture conditions that brings them down to more like to the 15 millimolar, but still it's not as great as primary or HepRG cells. And if we look at the activation of the stellate cells, we do see that you can see a collagen and lysyl oxidase upregulated after APAP. So we have a 4 cell type organoid culture, but it's not as great as the primary mouse, for instance, compared to primary human that we make ourselves. So are these any good? We do sometimes get pieces of liver, and we do isolate stellate cells, LSIC, Kupffer cells, and hepatocytes from them. They make okay spheroids. We cannot keep them as long in culture. And if we look at 7-day-old primary liver spheroids, this is the dotted line here. These 7-day cultures at CYP3A4 levels, they're about as high as our iPS-derived cultures. Stellate cells, they are a bit more activated in these primary liver spheroids. And if we do an APAP, the IC50 is better than our iPS cultures, even though CYP3A4 is a bit lower. So we're close, but of course not, we cannot make primary livers yet. Then last, we did some, and this is actually an earlier paper where the lab of Catherine Verfeuilly made gels that reflected most the extracellular matrix of hepatocytes. They tested over 200 different conditions, and they called it the HapMat gel. So if you put them in here, the same cell types, more or less differentiated in the same way, you get bigger agglomerates of cells where you see that you have albumin expression, HNF4 alpha, CYP3A4 islands. You even get some bile duct formation with keratin 19 expressing cells. If you expose these cells to oleic acid, you can also see an increase in lipid accumulation. And you can block this partially by using obeticholic acid and elafibranor. They quantify this a bit more on the level of IL-6, for instance, and you can see that both OCA and elafibranor blocked IL-6 production in these spheroid cultures, or sorry, organoid cultures. Same for procollagen. So you can also put these 4 cell types in a gel. It's a bit more cumbersome, but you get more structure. And we're currently looking into how we can study inflammation in these more natural environment, because you have more self-assembly of cell types in these gels. So maybe show you how some other people do this, like the future of organoid research. It's like the GWAS in a dish, is the Takebe lab. They are now able to make iPS from many different donor fibroblasts at the same time, which carry different SNPs. They make a kind of stem cell progenitor that they can freeze. They can expand, differentiate them towards human liver organoids. And then they can even stimulate these to, for instance, cause steatotic liver disease, and do screenings for instance, identify new SNPs that cause a higher fatty acid uptake or more lipid accumulation in these spheroids, and then do even genetic associations, because they have so many different iPS-derived lines. And if you're interested, please go to this Cell paper. It's really impressive what they did, just expanding the formation of this hepatocyte-like organoids and identifying, for instance, PNPLA3, which is a SNP associated with higher fat accumulation, but they also identified some new ones. So probably the world, well, those people working on organoids will go more and more into these huge batteries of iPS-derived cells, and either make these liver organoids. It's all in gels though, so that's maybe a bit less, especially the start is all in gel. But in the end, they also end up with spheroids, and probably this will be the next step, getting into the mechanism, working with all these different SNP, these different mutants, and so we can really form a city in a dish or a population in a dish, and look at really what toxic compounds do to different people, which in the end actually is what we want. And instead of looking at the HepG2 cells that become toxic, or one cell type that becomes fibrotic, we want to look at population-based studies. So I guess my time is more or less up. Just some take-home messages. I think liver organoids are suitable for disease modeling. Human primary, definitely, but it's very difficult to get them reproducible. Mouse is a very good alternative, but of course we would like to go for less mice and not more mice. Although from one mouse we can make anywhere between 500 and 700 spheroids, so it would mean that you could test many compounds in one from one mouse. iPS derived is very powerful. It's you can genetically modify them, but of course they're less mature than a human primary. What I did not talk about is that also people make ductal, so let's say the clavers-like organoid cultures. You can also make these also with matrigel, etc. You can have genetic modification as well, but for the moment, you can mainly have ductal and hepatocyte-like cells and not yet, for instance, stellate cells in there. Can we model MASLD and fibrosis? I think we can, yes, but we probably need to generate better stem cell-derived like cells, and especially MPCs. There's many people focusing on hepatocytes and making these better. But I think there's still a lot of room for improvement for good macrophages, Kupffer cells, good endothelial cells, and also even better stellate cells. What we also need is some vasculature in there, because there's of course no flow in these inside these spheroids. We could maybe go for chips. There's quite some companies out there that make hardware to seat cells on top of each other, or separated by a membrane, also supporting flow. I think that's really something to go for, although these systems are still pretty expensive. And we would like to incorporate more cells or tissues. The link between adipose tissue and liver is of course one of the few that would be very interesting. Also integration of more different immune cells, or challenge the exposure together with immune cells, would also be very interesting. Now I'll finish with thanking all my collaborators, funding agencies, and the people from my lab. So Inge Mannaerts here, she was at the basis of many of these co-culture experiments and the YAP work. Together with Lianne and Laura, they did, she did the mouse spheroid studies. The IPS cultures are done with, again, by Laura, but also Mina and Ayla. And IPS liver in gels are done with by Burak. And I thank you for your attention. And if there's any questions, I'm happy to answer them, if I can.
Thank you so much, Leo. It was great talk.
Thank you.
A lot of really good data, impressive data, I guess. I think.
Yeah, it's nice to see it maybe all together. I know people have, some of those papers people have seen, but to present it like this is maybe interesting for some people.
It already makes a nice story, isn't it?
Yeah.
I don't know if anyone has any questions. You can raise your hand. Anyway, I would like to ask you actually a couple of questions, or I mean, we are still not there with regarding the maturity of the cells, of the hepatocytes, especially that we derived from the iPSCs.
Yeah.
We are trying a lot of approaches, co-culturing to see if they mature. So the structure of the system, many approaches actually we can use now. It's not maybe a questions, but to know a little bit more about your thoughts on what should be our efforts. Do you think we have to improve our differentiation protocols, or it's really about the structures, or really about the cellular environment?
It's very difficult.
The $1 million question.
Yeah, if I see some of these papers by the Takibi lab, it's amazing what they do. They well, of course, it all depends what you want to do. If you want to look at fat accumulation and look at, discover SNPs or pathways that are involved in that, I think these hepatocytes that he makes are very good. If you want to go for toxicity studies, I guess what you want is to have a predictive system that, even if they're a bit less good than the hepatocytes that we have, or that you could have, like primary human hepatocytes or something, even if it's always constant and maybe tenfold less than a human hepatocytes, but if you know that, I would I would still like to think that that's a good system, right? It's what you need is good reproducibility and a system that can metabolize most of the drugs that you throw at them.
I'm sorry.
I don't know what that was. That you throw most of the drugs that they can metabolize most of the drugs that you throw at them. So you would like to have, you know, all the pumps and CYP enzymes there, which we don't have yet. So structure, yes, but I don't know what it would do. I think that for really long-term cultures, that might do the trick. Although if you use small spheroids, nutrients would get also pretty well to all the cells, even in the center. So it should not be a problem. But for instance, in our culture system, we don't have that many hepatocytes. So looking at biocanalically is more difficult because there's not all hepatocytes. That's a down part from us. So if you would have layered structure of hepatocytes and you could somehow put them on top, you know, put LSEC and stellate cells on top of them, your biocanalically formation would be probably much better. And so that would be easier to study there. Or if you had like these, you know, vessel-like hepatocyte-like structures, that would also be okay, which has been done as well. So it all depends what you want to study, and maybe we need like 3-4 different systems to cover everything.
Yeah. It depends a little bit on the objective, of course, of the study. And I also have another, it's a technical question. So when we do the RNA-seq from the spheroids, how can we distinguish the different cell types?
That's it's okay. I mean, if you do IPS, it's actually not that simple because you always have to compare with, well, you can plot it onto a single cell data set, but the hepatocytes from a healthy liver, of course, are not your cells. So you can never do that. If you do bulk sec, you can deconvolute, but again, you will need to have markers that represent your iPS-derived stellate cells or hepatocytes or endothelial cells. What people now do is they do nuc-sec. They dissociate the spheroids because they're very difficult to dissociate if you have them for 20 days. At least we don't manage very well. But you could do nuc-sec. We're now setting it up but we lose a lot of cells. But if the ones that you are left with are representative for the start material, I would think you could use nuc-sec. We're trying to work this out as well. It doesn't seem to work very easily. We cannot do it for primary, and we cannot do single cell on spheroids from iPS derived either. It's just, you know, it's too tough after 20 days or 14 days.
Yeah, we have the same problem. So,
but nuc-sec should work.
Okay,
I think.
So I guess Michelle has a question. Michelle?
Yeah, I have. Unfortunately, I can't turn on my camera. Apparently, it's blocked by you or by the organizer. But first of all, thank you very much, Leo, for your very interesting talk. I was just wondering, you mentioned when you were reconstituting the 4 cell types, and you put them in the stoichiometry like in vivo, they don't form quite well. Those organoids, the spheroids.
Yep.
Can you elaborate on that? Why that, in your opinion, is not occurring? It might actually then indicate that there's something missing there still, right?
Yeah, it could be that there's something missing, but it's so the way we form them is we put the cell suspension together, and then we just with a small pipette handler thingy. We put them just 10 microliters at the bottom of a well, or 10 to 50 microliters at the bottom of a well. We let it sit for a while, and only the next day we add more medium. So we try to create the best circumstances that they will aggregate, but they just don't aggregate. We could play around, of course, with, for instance, adding collagen to it, or even more serum, because we do the spheroid formation, we do in serum, and afterwards we remove all serum.
Right.
Yes, I think we could play with that, but yeah, It's
This is also occurring with the work that you're collaborating with, where they use these polymers, the ECM mimicking material.
Yeah, of course, there's not a problem because you lock them up in the gel.
Okay.
Then you get islets, and they use a lot more gels, a lot more cells, and you get islets in this gel where they, and cells are going in, kind of move around. It's a completely different setup, of course. I think if we put just Matrigel, it will all survive, and you get a gel where all the cell types are in, and probably in the good ratio at the start at least.
In this polymer, they are still proliferating or not?
In which one? In the spheroid or in the gel?
In the gel.
In the gel, what we have is that the way we did it at the start, this first paper, is we still use MCSF, so the monocytes are still proliferating.
Okay.
The other cells, after a while, they do not proliferate anymore, and which is more or less the same in our spheroids, but there we don't use the monocyte growth factors, because if you do that, you'll get blebs, coming off of the spheroids, which are probably the monocytes that start to proliferate, and the rest doesn't.
Yeah,
So
I was making this question, maybe it doesn't depend in which stoichiometry you start them with in the polymer. If they are still proliferating, maybe they gonna adapt to the stoichiometry, which is more close to the in vivo situation. That was my idea.
Could be. But as you can imagine, probably they're easy experiment, but they last too long to really try a lot. I mean,
Yeah,
in the gels, it's 40 days. Actually, it's horrible to do these experiments because you cannot do many experiments on a year, right?
No, it's very time consuming. I know.
So I guess we could try a lot of different conditions. It's just you have to have quite some time, hands, and money to do it.
Right. Okay, thanks.
Yeah.
Thank you so much, Michel, Leo. Bernard, I guess you also have a question.
Yes, actually, I have two questions. First of all, a very interesting talk. My first question is about acetaminophen that you use to induce fibrosis. However, this drug is not known as a prototypical drug inducing fibrosis in human. But I wonder whether, whatever your reason, but I wonder whether you tried other pro-fibrogenic drugs such as amiodarone, paraxylene, or toxic compound like carbon tetrachloride in your model.
Yeah, good question. And we get this question every time we submit something. So the APAP is actually very interesting. There's now a couple of papers. The first paper we had the same question from the reviewer. Maybe it was you. No, I don't think so.
No, it wasn't. It wasn't.
We had the same question. APAP. Nobody gets fibrosis with APAP. We tested it in mice, and if you just give a mouse 4 weeks low doses APAP, it gets fibrosis. It was just to use it as a damaging agent to hepatocytes that then we could get stellate cell activation. We cannot use CCL4 because CCL4, it goes everywhere, and then your entire incubator is full with CCL4. We cannot really use it. Else we would have used CCL4, of course. It would be much, much easier. We used TAA as well, and methotrexate as well, and you can have also the same activation, especially if you do long-term exposure. You can have fibrosis in these spheroids.
Okay, and my second question, if I may, is related to the MASLD model. And you try different drugs in order to reverse the fibrosis, and I wonder whether you tried the first drug ever approved by the FDA, which is resmetirom. Yeah. And what about? Do you plan to do that or?
Yeah. We bought it a while back, and we're definitely gonna test it. Yeah, sure.
Okay.
Of course.
Okay, and in the next step, it would be interesting to try different iPSC from donor with different PNPR3 variant to see if this drug can be efficient in the different variants. Because
Absolutely.
I think it could be expected that the effect would not be the same depending of the variants. So I mean, I think that you will have a very interesting result if you can keep on this subject.
Yeah, absolutely. So what we and this is all not published. We're busy actually. Well, we're busy, but we might not be able to finish this work. We have human ES cells, which are homozygous for the PNPLN3 variant. We have also human ES cells that are homozygous for the HSB17B3. We have heterozygous for each, and we're testing them for hepatocyte toxicity, lipid loading, but also stellate cell activation. And we can make an entire spheroid, of course, PNPL positive or negative, blah blah blah.
Okay.
Or only the stellate cells, but it's again, it's a lot of cultures and
I know we are doing also some organoids in the lab. So I mean, but not with iPSC, but it's time consuming and
sometimes things don't work out the way you want, etc. Right?
Yeah, and thank you very much for your presentation and the answer.
You're welcome.
Okay, thank you. I don't think we have more questions, at least it doesn't look like. Again, Leo, thank you so much for joining us today. And if there are no more questions, I thank you all as well for attending the seminar.
Okay.
And see you all soon. Thank you.
Bye-bye.
Bye-bye.
Nice seeing you.
Thank you very much for the presentation. Yeah.
Bye. Bye.
Bye-bye.